Hello, and welcome to Local Adaptive Deep Learning in the Brain, a study on the biological plausibility of bursting cortico-cortical networks. In the brain, neurons receive signals through their basal dendrites. This determines the somatic potential, which can be seen at the bottom as VL. These signals are then used to determine the firing rate of the soma, or the cell body. This is interpreted as being done through an activation function. In our case, this is a sigmoid function. This firing rate is then projected along the axon to neurons in the next layer, with synapses determining how the signal is adapted for each neuron. To adapt synaptic weights in response to error, the brain must be capable of attributing this error to individual synapses. This involves sending error signals backwards across many layers. Artificial neural networks solve this problem through backpropagation. Whilst backpropagation is plausible in the output layer, earlier layers require that the error signal be sent backwards along feed-forward weights. This is not plausible. So an alternative is to project error signals along separate connections. Pyramidal cells allow this, so they have both basal and apical dendrites, which makes them capable of receiving top-down signals into the apical dendrites and bottom-up signals into the basal dendrites separately. However, it's thought to be biologically impossible for one connection to have knowledge of another's weight, and so Y can't adapt to match W. This is known as the weight transport problem. But Y doesn't need to have the exact same value as W, it just needs to have the same sign. Though the signal may be of different magnitudes, as long as the weights still adjust in the correct direction, learning still occurs. Feedback alignment approximates the performance of backpropagation through attributing random frozen values to Y. The feed-forward weights in this model adapt to send meaningful error signals through Y, thus allowing learning to occur. Burst CCN is a model built for approximating backpropagation in the brain, and is built upon the theory of burst-dependent learning. In simple terms, in this model, the error attributable to a neuron is communicated through the variation in its burst rate about the baseline. For neurons in the previous layer to decipher the error attributable to this neuron, both the event rate, which is its activated potential, and so like the forward signal it sends, and the burst rate need to be communicated backwards. Short-term facilitating weights, which are the orange Y weights, communicate the burst rate from this soma backwards through a somatostatin or SOM producing Martinotti interneuron, whilst short-term depressing weights, which are blue and are the Q weights, communicate the event rate directly. These signals are received in the apical dendrites and are used to determine the burst probability and thus the variation in the burst rate about the baseline. First, CCN is quite a new model, so we want to determine whether pyramidal neurons and the SOM interneurons through which the burst rates are fed back behave in the same way as their real-life counterparts. In order to achieve this, I studied 4 models, but for simplicity, I will just discuss 2 in this presentation. The first is the feedback alignment model of burst CCN, which has random frozen feedback weights through Y. This is the most plausible model, as it involves no weight transport problem. Therefore, alignment with actual real neuron behavior would suggest that this model is accurate. I also studied a model with Y tied to the weight between the current pyramidal neuron and the next. This is the model that most closely approximates backpropagation, but suffers from the weight transport problem. Therefore, alignment with actual real neuron behavior would suggest that the brain has found a way of solving the weight transport problem. The experiment I compared these burst CCN models to was by Kahn et al., which studied the relationship between the behavior of and relationship between pyramidal cells and interneurons in layers 2 and 3 of V1 in the visual cortex of mice in response to a learning task. One of the interneuron classes was the SOM producing class. This represents the Y interneuron in the burst CCN model. This study therefore focuses on the behavior of pyramidal cells and the SOM cells. In this experiment, mice were run through a virtual reality corridor, as shown in the picture, before being presented with either vertical or angled grating. The mouse received a reward for licking a spout in front of them when presented with the vertical grating, but no such reward for licking in response to the angled grating. and all mice learn to distinguish between the two stimuli. In my experiment, I presented each of the models, each of the burst CCN models, with either vertical grating, which had a target value of 1, or angled grating with a target value of 0. The first comparison I made between the burst CCN models and the real neurons was in absolute selectivity change. So selectivity is the mean firing rate of a cell in response to the vertical grating minus the mean firing rate in response to the angled grating. Therefore, the change in the absolute selectivity can be interpreted as the change in the difference in a cell's response between the two stimuli. So you would expect that with learning, the difference gets bigger. Both the event rate and the burst rate, which are the two parameters that contribute towards the pyramidal firing rate, increase their selectivity. This replicates the behavior seen in Carnet al. For both the feedback alignment models and the backprop model, these results were the same. As you can see in Carnet al, there is an increase in the absolute selectivity with learning in some interneurons, though this is not significant, and this is again replicated in both models studied of burst CCN. The second comparison I made between the burst CCN model and the findings of Carnet al is in the selectivity correlation change. So this shows the relationship between the selectivities of some interneurons and local pyramidal cells both before and after learning. So you can see in the burst CCN results on the left, the feedback alignment model sees a positive correlation between the burst rate and the SOM interneuron selectivities prior to learning. And this relationship improves with learning. This aligns with the observations of Carnet al, which you can see on the right, where the R improves from 0.38 to 0.51. In the burst CCN backpropagation model, a similar trend is also seen, but it's seen in between the event rate and the SOM interneuron through Y, as well as through the burst rate and the SOM interneuron through Y. Al's next finding was that pyramidal cells that had a low stimulus-independent correlation with SOM interneurons saw significantly higher increase in absolute selectivity with learning, which you can see on the left. This finding is replicated in the first hidden layer of the burst CCN feedback alignment model. Again, this is the leftmost local finding. However, the same relationship in the burst CCN feedback alignment model is found with other pyramidal neurons in their local area, which is not replicated in Carnet al. Conversely, the backpropagation model of burst CCN sees completely the inverse relationship, with higher coupling meaning higher absolute selectivity change. This is the opposite result to which was seen in Carnet al. In summary, both models, in line with the real neurons seen in Carnet al, experience significant increases in absolute selectivity of pyramidal cells and SOM interneurons with learning. Both models also see selectivity correlations that align with those seen between real pyramidal cells and real SOM interneurons. While the feedback alignment model sees higher learning in pyramidal neurons less coupled with SOM neurons in the first hidden layer, again aligned with real pyramidal neurons, they see the same relationship with other pyramidal neurons, which is not seen in real pyramidal neurons. The backpropagation model, however, sees higher learning in pyramidal cells that are more coupled with SOM neurons, which is completely unaligned with real neurons. Limitations of my methodology do, however, exist. For example, the task I've created could be perceived as identifying vertical and angled gratings, while if a lot of other images were added with the same target value as the angled grating, this could be turned into a task of just identifying the reward stimulus. Also, the classic role of the Martinelli interneuron, which is the SOM interneuron, is in inhibiting the pyramidal cell, which is the opposite to the role it occupies in the burst CCN model. A reinterpretation of this role could actually reverse the relationship between pyramidal and SOM coupling seen in the backpropagation model, and so have it better align with the results seen in Carnet al. Thank you for listening.